Now we are going to learn basic structure of antibodies. Recognition of an immunogen by the surface antibodies of B cell triggers proliferation and differentiation into memory B cells and plasma cells. Plasma cells secrete soluble antibody molecules with specificity for antigen that is identical to the surface receptor of the parent B cell. So, recognition of an immunogen by the surface antibodies of the B cell triggers proliferation and differentiation into memory B cells and plasma cells. So, plasma cells secrete soluble antibody molecules with specificity for antigen that is identical to the surface receptor of the parent B cell. The following section will describe the structural features of the antibody molecule that allow them to fulfill their two major functions. Binding foreign antigens encountered by the host, mediating effector functions to neutralize or eliminate foreign invaders. So, binding foreign antigens encountered by the host. Then mediating effector functions to neutralize or eliminate foreign invaders. It has been known since the late 19th century that antibodies reside in the blood serum. In the mid 20th century, antibodies were shown to be present in other secreted body fluids, milk, tears, saliva, bile, etc. Blood can be separated in the centrifuge into a fluid and a cellular fraction. This fluid fraction is the plasma, and the cellular fraction contains red blood cells, leukocytes, and platelets. Plasma contains all of the soluble small molecules and macromolecules of blood, including fibrin and other proteins required for the formation of blood clots. If the blood or plasma is allowed to clot, the fluid phase that remains is called serum. The first evidence that antibodies were contained in particular serum protein fraction came from a classic experiment by Arne Theselius and Elvin A. Cabot in 1939. They immunized rabbits with the protein ovalbumin, the albumin of egg whites, prepared serum, and then divided the serum into two aliquots. Electrophoresis separation of one serum aliquot revealed four major fractions corresponding to serum albumin and the alpha, beta, and gamma globulins. Prior to electrophoresis, the other serum aliquot was mixed with immunizing antigen ovalbumin, allowing formation of an immune precipitate complex of antigen and antibody that was removed. So, leaving the remaining serum proteins which did not react with the antigen, a comparison of the electrophoretic profiles of these two serum aliquots revealed that there was a significant drop in the gamma globulin peak in the aliquot that had been reacted with antigen. Thus, the gamma globulin factor was identified as containing serum antibodies, which were called immunoglobulins to distinguish them from any other proteins that might be contained in the gamma globulin fraction. The early experiments of Cabot and Theselius resolved serum proteins into three major non-albumin fractions, alpha, beta, and gamma. We now know that although immunoglobulin G, IgG, the most abundant class of antibody molecules, is indeed mostly found in the gamma globulin fraction, significant amount of it and other important classes of antibody molecules are found in the alpha and beta fractions of serum. Then antibodies are heterodimers. Antibody molecules have a common structure of 4 peptide chains. This structure consists of 2 identical light chains, polypeptides of about 22,000 Dalton, and 2 identical heavy chains. Larger polypeptide of around 55,000 Dalton or more. Each light chain is bound to a heavy chain by a disulfide bond and by non-covalent interactions such as salt linkages, hydrogen bonds, and hydrophobic interaction to form a heterodimer. Which bonds are there? Disulfide bond, and by non-covalent interactions such as salt linkages, hydrogen bonds, and hydrophobic interactions to form a heterodimer. Similar non-covalent interactions and disulfide bridges link the two identical heavy and light chain combinations to each other to form the basic 4-chain antibody structure, a dimer of dimers. As we will see, the exact number and precise position of the disulfide bonds linking dimers differs among antibody classes and subclasses. The first 110 or so amino acids of the amino terminal region of a light or heavy chain varies greatly among antibodies of different antigen specificity. These segments of highly variable sequence are called V regions, VL in light chain and VH in heavy. All of the differences in specificity displayed by different antibodies can be traced to differences in the amino acid sequence of V regions. In fact, most of the differences among antibodies fall within areas of the V region called complementarity determining regions or CDRs. And it is these CDRs or complementarity determining regions on both light and heavy chains that constitute the antigen binding site of the antibody molecule. By contrast, within each particular class of antibody, far fewer differences are seen when one compares consequences throughout the rest of the molecule. The regions of relatively constant sequence beyond the variable region have been dubbed C regions, Cl in light chains and Ch in heavy. Antibodies are glycoproteins. With few exceptions, the site of attachment for carbohydrates are restricted to the constant region. We do not completely understand the role played by glycosylation of antibodies, but it probably increased the solubility of the molecule. Inappropriate glycosylation or its absence affects the rate at which antibodies are cleared from the serum and decrease the efficiency of interaction between antibody and other proteins with which it interacts. So when we try to look into the schematic diagram of structure of immunoglobulins derived from amino acid sequence analysis, you could see a structure like this. This is schematic diagram of structure of immunoglobulins derived from amino acid sequence analysis. So each heavy and light chain in an immunoglobulin molecule contains an amino terminal variable region, aqua and tan respectively, that consists of 100 to 110 amino acids and differ from one antibody to the next. The remainder of each chain in the molecule, the constant C regions, purple and red in this picture, purple and red, exhibit limited variation that defines the 2 heavy chain subtypes and the 5 heavy chain subclasses. Some heavy chains also contain a proline rich hinge region. The amino terminal portions corresponding to the V regions bind to antigen. Effector functions are mediated by the carboxy terminal domains. The mu and epsilon heavy chains, which lack a hinge region, contain an additional domain in the middle of the molecule. CHO denotes a carbohydrate group linked to the heavy chain. So NH3 plus regions, NH3 plus regions 4, then VL, V heavy, Cl. V in the sense variable domain, okay, variable conserved domains 4 sort of. Hinge region, antigen binding, effector activity, then light chain, kappa or delta, heavy chain, mu, gamma, alpha, delta or epsilon. Once again we are going to read this. So each heavy and light chain in an immunoglobulin molecule contains an amino terminal variable region V, aqua and tan respectively, that consists of 100 to 110 amino acids and differs from one antibody to the next. The remainder of each chain in the molecule, the constant C regions, purple and red, exhibits limited variation that defines the 2 light chain subtypes and the 5 heavy chain subclasses. Some heavy chains, gamma, delta and alpha, also contain a proline rich hinge region, black region. The amino terminal portions corresponding to the V regions bind to antigen. Effector functions are mediated by the carboxy terminal domains. The mu and epsilon heavy chains which lack a hinge region contain an additional domain in the middle of the molecule. CHO denotes a carbohydrate group linked to the heavy chain. Then chemical and enzymatic methods revealed basic antibody structure. So our knowledge of basic antibody structure was derived from a variety of experimental observations. When the gamma globulin fraction of serum is separated into high and low molecular weight fractions, antibodies of around 150,000 Dalton, designated as immunoglobulin G (IgG), are found in the low molecular weight fraction. In a key experiment, brief digestion of IgG with the proteolytic enzyme papain produced 3 fragments, 2 of which were identical, with a third that was quite different. The 2 identical fragments had antigen binding activity and were called Fab fragments. Fragment antigen binding. Okay, Fab fragments. The other fragment, 50,000 Dalton, had no antigen binding activity at all. Because it was found to crystallize during cold storage, it was called the FC fragment. Fragment crystallizable. Then digestion of IgG with a different proteolytic enzyme pepsin demonstrated again that the antigen binding properties of an antibody can be separated from the rest of the molecule. Wait a minute, I will show you a picture. Yeah. Prototype structure of IgG showing chain structure and interchain disulfide bonds. The fragments produced by enzymatic digestion with pepsin or papain or by cleavage of the disulfide bonds with mercaptoethanol are indicated. Light chains are in grey and heavy chains in blue. Here you could see disulfide bonds are there. L chain is there, H chain is there. Then pepsin digestion, we could see FC fragments. Then pepsin digestion, Fab regions. Mercaptoethanol reduction, we could see some other regions. Okay. So we were learning about fragment crystallizable region. So digestion of IgG with a different proteolytic enzyme pepsin demonstrated again that the antigen binding properties of an antibody can be separated from the rest of the molecule. So pepsin digestion generated a single 100,000 Dalton fragment composed of 2 Fab-like subunits designated the FAB/2 fragment which binds antigen. The FC fragment was not recovered from pepsin treatment because it had been digested into numerous small peptides. A key observation in reducing the multi-chain structure of IgG was made when the molecule was subjected to mercaptoethanol reduction and alkylation, the chemical treatment that irreversibly cleaves disulfide bonds but not peptide bonds. If the post-treatment sample is chromatographed on a column that separates molecules by size, it becomes clear that the intact 150,000 Dalton IgG molecule was in fact composed of subunits. So each IgG molecule contains 2 larger polypeptide chains of around 50,000 Dalton designated as heavy chains and 2 smaller chains of about 22,000 Dalton designated as light chains. Then antibodies themselves were used to determine how the enzyme digestion products Fab-2 and FC were related to the heavy chain and light chain reduction products. This question was answered by using anti-sera from goats that had been immunized with either the Fab fragments or the FC fragments of rabbit IgG. The antibody to the Fab fragment could react with both the H and L chains, whereas the antibody to the FC fragment reacted only with the H chain. These observations led to the conclusion that the Fab fragment consists of a portion of a heavy plus intact light chain and that FC contains only heavy chain components. From these results and those mentioned about the structure of IgG shown was deduced. So according to this model, the IgG molecule consists of 2 identical H chains and 2 identical L chains which are linked by disulfide bridges. The likely next step in developing a picture of antibody structure was determining their amino acid sequence, yet here a problem arose. The problem the population of antibodies in the serum alpha globulin fraction consists of a heterogeneous spectrum of antibodies, many different antibodies each present in small amounts. Even if immunization is done with a hapten carrier conjugate, the population of antibodies raised against the hapten is heterogeneous because multiple antibodies recognize different epitopes of the hapten. Then amino acid sequence analysis, however, requires a pure sample of the molecule under study. This obstacle to sequence analysis was overcome by the use of immunoglobulins from patients with multiple myeloma, a cancer of antibody producing plasma cells. The plasma cells is a normal individual are end stage cells that secrete a single molecular species of antibody for a limited period of time and then die. In contrast, a clone of plasma cells in an individual with multiple myeloma has escaped normal controls on their lifespan and proliferate in an unregulated way without requiring any activation by antigen to induce proliferation. Although such a cancerous plasma cell called a myeloma cell has been transformed, its protein synthesizing machinery and secretory functions are not altered. Thus, the cell continues to secrete molecularly heterogeneous antibody. This antibody is indistinguishable from normal antibody molecule but is called a myeloma protein to denote its source. In a patient afflicted with multiple myeloma, so myeloma protein can account for 95% of the serum immunoglobulins. In many patients, the myeloma cells also secrete excessive amounts of light chains. These excess light chains were first discovered in the urine of myeloma patients and were named Benson's proteins for their discoverer. So, multiple myeloma also occurs in other animals. In mice, it can cause spontaneously, as it does in humans, or conditions favoring myeloma induction can be created by injecting mineral oil into the peritoneal cavity. The clones of malignant plasma cells that develop are called plasmacytomas, and many of these designated MOPs denoting the mineral oil induction of plasmacytoma cells. A large number of mouse MOPC lines secreting different immunoglobulin classes presently carried by the American Type Culture Collection, a non-profit repository of cell lines commonly used in research. The method for producing monoclonal antibodies of a desired specificity pioneered by Georges Kohler and Cesar Milstein makes use of the plasmacytoma lines. This hybridoma technology allows production of homogeneous antibodies for structural and functional studies and for a variety of clonal applications. Then, light chain sequences revealed constant and variable regions. When the amino acid sequences of several Bence-Jones protein light chains from different individuals were compared, a striking pattern emerged. The amino terminal half of the chain, consisting of 100 to 110 amino acids, was found to vary among different Bence-Jones proteins. So, light chain sequences revealed constant and variable regions. When the amino acid sequence of several Bence-Jones proteins, that means light chains, from different individuals were compared, a striking pattern emerged. The amino terminal half of the chain, consisting of 100 to 110 amino acids, was found to carry among different Bence-Jones proteins. This region was called the variable V region. The carboxyl terminal halves of the molecule, called the constant C region, had one of two amino acid sequence. This led to the recognition that there were two light chain types, kappa and lambda. In humans, 60% of the light chains are kappa and 40% are lambda, whereas in mice, 95% of the light chains are kappa and only 5% are lambda. A normal antibody molecule contains only one light chain type, either K or lambda, never both. The amino acid sequence of lambda light chain consist regions show minor differences that are used to classify lambda light chains into subtypes. In mice and in humans, there are 4 subtypes: lambda 1, lambda 2, lambda 3, and lambda 4. Amino acid substitutions are only a few positions are responsible for the subtype differences. There are 5 major classes of heavy chains. For heavy chain sequence analysis, myeloma proteins were reduced with mercaptoethanol and alkylated, and the heavy chains were separated by gel filtration in a denaturing solvent. When the amino acid sequence of several myeloma protein heavy chains were compared, a pattern similar to that of the light chains emerged. The amino terminal part of chains consisting of 100 to 110 amino acids showed great sequence variation among myeloma heavy chains and were therefore called the variable region. The remaining part of the protein revealed 5 basic protein sequence patterns corresponding to 5 different heavy chain constant regions: mu, delta, gamma, epsilon, and alpha. Each of these 5 different heavy chains is called an isotype. So, the length of the constant region is approximately 330 amino acids for delta, gamma, and alpha, 440 amino acids for mu and E. The amino acids chains of a given antibody molecule determine the class of that antibody: IgM, IgG, IgA, IgD, IgE. H chains of each class may pair with either kappa or delta light chains. The single antibody molecule has 2 identical heavy chains and 2 identical light chains. Minor differences in the amino acid sequence of the alpha and gamma heavy chains lead to further classification of the heavy chains into subisotypes that determine the subclass of antibody molecules they constitute. In humans, there are 4 subisotypes of a heavy chain: alpha 1 and alpha 2, and thus 2 subclasses, IgA1 and IgA2, and 4 subisotypes of gamma heavy chains: alpha 1, gamma 2, gamma 3, and gamma 4, and thus 4 subclasses: IgG1, IgG2, IgG3, IgG4, and in mice there are 4 subisotypes: alpha 1, alpha 2A, alpha 2B, alpha 3, and the corresponding subclasses.